Good morning. Today we are going to take the remaining part of our sixth chapter, Unraveling Genetic Mysteries. So, last class or previous class, we have studied about the genetic structure of DNA and then about gene, how gene action or gene acting for the production of protein, and all those things we have studied in our previous class. So today we are going to study about the variation, the reason for variation. There are certain reasons for the variation. Among those reasons, one of the reason is mutation. Mutation means sudden heritable changes in the genetic constitution of an organism is known as mutation. Mutation can be caused due to any defect in the gene or maybe due to the radiation or due to the action of certain chemicals or due to any changes in the genetic constitution. All these things are responsible for the mutation. So once more I will repeat for you. Mutation means sudden heritable change in the genetic constitution of an organism is called as a mutation, and this sudden heritable change result in the variation. And the reason for this mutation is radiation, certain action of certain chemicals, or changes in the genetic constitution. All these things are responsible for the mutation. And another thing responsible for variation is first point is mutation. Second, you are going to study about is crossing over. Now we can see the certain steps in the crossing over. Okay. So you can see in the picture, two chromosomes are shown. After that, that's in the first stage. In the second stage, you can see two chromosomes are mixing together. And in the third stage, you can see that two chromosomes exchange their parts each other. So both chromosomes characters exchanged, and a new character, a chromosome with a new character is formed in the third stage. So during the initial phase of meiosis and mitosis you have studied in the 9th standard. So during the initial phase of meiosis is the division of sex cells. So during the initial phase of meiosis, chromosomes pair and exchange their parts. Okay, so we were discussing about the reasons for the variation. Mutation we have already told. And about the crossing over also told. That means in crossing over during the initial phase of the meiosis, chromosomes exchange their parts. And when the chromosomes exchange their parts, the newly formed chromosome will be having a different genetic constitution. And that result in a new character, that result in the variation in the offspring, and new character, formation of a new character in the offspring. So mutation, then crossing over, and another reason for the variation is the fertilization, at the time of fertilization. So from the parents to the offspring, the characters are transferred from parents to offspring through the gametes. Gametes means sex cells. So when this gametes, fusion of the gametes take place, that means during fertilization, male gamete and female gamete fuses together. At that time, what happened? That allele, the allele will be exchanged. That means a new allele combination will be formed. That is from the father and from the mother, the characters join together, and a new character will be formed during the fertilization. A new character will be a new genetic constitution will be formed. A new allele combination will be formed for the offspring, and that results in the variation. And during the starting of our chapter, we have told that the characters that are different from our parents is known as a variation. That is a variation. And the characters that we get from our parents, that is transmitted from our parent to offspring, and that is the same character when it is transmitted from parent to offspring, that is heredity. And the character that is different from our parents, that is variation. And the reason for variation and what are the chances for causing the variation is mutation, crossing over, and fertilization. It's clear for you. And during fertilization, from the parents, the characters are transmitted through the gametes. And during the fertilization, male and female gametes fuses together, and a new genetic constitution will be formed, and a new character will be formed for the offspring, and that will result in the variation. This is clear for you. Now we are going to see about how male and female child is born. That we can see now. How we can. Here we have already discussed about the number of chromosomes present in the human beings. Last class we have discussed that the 46 number of chromosome is present in our human cell. And in that 44 chromosomes are somatic chromosomes and 2 chromosomes are sex chromosomes. Okay, so for the female, 44 plus XX is the genetic constitution, and for male, 44 plus XY is the genetic constitution. So in female, the 2 sex chromosomes present is the 2 X chromosome. In male, 2 sex chromosome present is 1 X and 1 Y chromosome is present. During the time of fertilization, when X chromosome from mother fuses with the X chromosome from father, the child born will be female. Okay, so when the X chromosome from mother fuses with the X chromosome from father, the child born will be female because the offspring will be having the genetic constitution 44 plus XX. Then when the X chromosome from mother fuses with the Y chromosome from father, the child born will be male, and the genetic constitution will be 44 plus XY. So you should keep in mind that genetic constitution of the father determines the sex of the newborn child. Is it clear for you? So the genetic constitution of the male is 44 plus XY, and genetic constitution of the female is 44 plus XX. When the X chromosome from mother fuses with the X chromosome of father, the child born will be female because it will be having a genetic constitution 44 plus XX. Then when the X chromosome from mother fuses with the Y chromosome from father, the genetic constitution of the child will be 44 plus XY. Then the child born will be male. So the genetic constitution of the father determines the sex of the child. Is it clear for you? Then next is the skin color. We know that we are seeing the people with the various skin color. The skin color is some will be fair, some will be dark. One of the reasons for that is the presence of melanin. Melanin is the pigment. If the melanin content is more in our body, we will be dark in color. If it is less in our body, we will be fair in color. And that melanin pigment concentration is controlled by a gene in our body. So that gene controls the presence of melanin pigment in our body. So according to that, we are having our skin color will be fair or dark. Then another thing is, according to the environmental condition, we are having the skin color. In different countries, the skin color is different because in different countries, the different environment is clear for you. So according to that, as an adaptation to the environment, we are having the skin color. So the skin color is controlled by the presence of melanin pigment in our body, and that melanin pigment and the concentration of this melanin pigment is controlled by a gene. So according to that, our skin color is fair or dark. Is it clear? Okay, so by this topic, our 6th chapter is finished, and the 7th chapter is a continuation of this 6th chapter. Because in this 7th chapter, we are going to study about the how the genetics is applicable in our daily life, or how it is applicable for the future generation. So next is the genetic of the future. So our seventh chapter name is genetics of the future. It's a continuation of this chapter. Okay. So seventh chapter name genetics of the future. In this chapter, we are going to study about genetic engineering, biotechnology, all these things. Biotechnology means making use of the microorganisms in our daily life or for our needs. Making use of these microorganisms. That technology, the technology of using these microorganisms, that technology is known as biotechnology. It's clear for you. And in genetic engineering, the basics, the trait of an organism or the character of an organism is controlled by removing the gene and replacing another gene instead of that. So in order to cut the gene and replace the gene, certain enzymes are used. All those things we will study. Before that, we have to see the production of artificial insulin. We have studied in our third chapter. What is insulin? Insulin is a hormone that regulates the amount of glucose in our blood, isn't it? So, if our blood, the production of insulin, hormone insulin is less, what happens? The person will become diabetic. So the presence of glucose, when the presence of glucose in our blood increases than the normal level, the insulin will be produced and it will convert that glucose into glycogen. It's clear for you. So we have studied about the function of insulin. That is normally, that naturally produced insulin in our body. But you have heard about for diabetic patient, if it is too much severe, they will be taking insulin injection. So that is taken by using the artificial insulin. So how this artificial insulin is produced? That we can see the steps of producing artificial insulin. We can see that now. So we can produce the artificial insulin. So here you can see the cell of a bacteria and a human cell is shown here. Both bacteria and human cell consist of DNA. The DNA of the bacteria is known as the plasmid. And then we are also having DNA. On the DNA, what will be present? Genes are present. So in our body, inside the cell, inside the nucleus, genes responsible for the production of insulin is present. So we are cutting the gene that is responsible for the production of insulin, and we are joining that gene on the plasmid of the bacteria. So you can see in the picture, cutting of insulin gene from the human DNA, and we are joining it with the gene of the bacteria plasmid. That plasmid is the DNA of the bacteria. Okay, so you can see in the picture and that color difference where the joined gene, that is insulin producing human gene is joined. You can see a color change in that. That joined gene, that DNA with the insulin producing gene, that DNA is inserted, again inserting that DNA into the bacteria. So when this new genetic constitution is inserted in a bacteria, that bacteria is now capable of producing insulin, because from the human, that gene that is capable of producing insulin is cut and joined, is pasted in the plasmid of the bacteria. So plasmid with the ligated means joined, ligated insulin, and ligated insulin gene is inserted into bacterial cell. Then bacteria that multiply in the culture medium produce inactive insulin. So in the laboratory, under certain specific condition, this bacteria is allowed to multiply. Then when this bacteria is allowed to multiply, that new bacteria is capable of producing inactive insulin, and we are converting that inactive insulin into active insulin. Okay. These are the steps in the genetic engineering. That is, with the help of genetic engineering, we are doing all these things. For this, we are using genetic scissor. For cutting the gene, we are using genetic scissor, and for inserting that gene, we are using genetic glue. Now we can see which is the genetic scissor and which is the genetic glue. Okay. So two enzymes are the genetic scissor and genetic glue. We can see that. That is restriction endonuclease, the enzyme used to cut DNA at specific site. This enzyme is known as genetic scissors. So keep in mind the genetic scissors is restriction endonuclease. Then enzyme ligase. The enzyme ligase is used for joining DNA at specific sites. This enzyme is called as a genetic glue. Then vectors. Vectors means carriers. Plasmids in bacteria, that is, plasmid means DNA in bacteria, are generally used as vectors or carriers. A gene from one cell is transferred to another cell by using suitable vectors or carriers. I think it's clear for you about genetic scissor and genetic glue. Restriction endonuclease is known as the genetic scissors, and the ligase, enzyme ligase, is known as the genetic glue. So restriction endonuclease, genetic scissors, and enzyme ligase, genetic glue. So keep in mind about this. I think it's clear for you. Remaining part we can take in the next class. Okay. Thank you.